I have a confession to make. I'm an NMR spectroscopist by training, so I did solve some structures, but they were always in solution. But that's not the topic of today, likely, and I'll just focus on metals in mainly protein structures, actually. Which are the focus of our work with this team of people. So Claudia is my main co-worker since a number of years. And she's the person who has designed and carried forward most of the ideas that I'm going to discuss with you today. Gabriele and Serena have contributed to developing the database and some of the tools that I will be showing together with Jana. And Valeria is the current postdoc who is doing most of the upgrades to the database in terms of front and back end. So metal ions we are the focus of our research, and that's because they are essential to living systems. We estimate, just by looking at PDB contents, that about 40% of enzymes, for example, require metals for their catalytic function. And of course, as you are probably aware of, the role of metal is not limited to sustaining catalysis. They're also involved in structural roles for keeping, for example, the three-dimensional structure with the proper fold. So we set out quite some years ago now to look just at the core of these metal binding structures, that is the metal binding site. And we focus on on this idea that we call the minimal functional site, that is we focus on the metal ion, and which is the center of our interest. And so that's, sorry, I don't get how. So we start, I'll replay. So we start from the metal ion and we look at the amino acids that are binding directly to the metal ion via coordination bonds. These are very straightforward to identify from a crystal structure in the PDB. And then we extend this substructure that we are looking at by taking into account all the amino acids that are making contact with any of the metal binding amino acids. So we expand from a minimal binding site, which is just the metal and the surrounding amino acids that have a coordination bond to the metal, to this minimal functional site, which is roughly a sphere centered on the metal, except it's not really spherical by construction. And that's what we have cataloged in the Metal PDB database. So the Metal PDB is an archive of all the metal sites in PDB, which of course we can extract automatically quite easily. And the Metal PDB website can be queried let's say Google style, just by typing in the PDB code of the structure you are interested in, or in a more complicated way with this advanced search table that allows you to browse for by protein name, by enzymatic activity, by metal, of course, and so on and so forth. So we'll just have a couple of examples to give you an idea of what it looks like. So 12CA is the structure of human carbonic anhydrase. It's a very famous zinc enzymes. And this specific structure actually contains two different metal sites that we both cover in the Metal PDB database. One is the physiological zinc site, and the other one is a spurious mercury binding site. So I only have the physiological one as an example, and there are some features that we extract from the database. So the domain composition, the amino acids that are coordinating the metal ion. And one of the things we've been working on recently is highlighted by the green circle here, and it's the manual annotation of the physiological relevance of that site. So this one says this site is physiological, and actually if you click on the small book icon, it will provide you with a link to a small selection of literature that supports the assignment of that site as a physiologically relevant one. This is another example. It's bovine superoxidase mutase, which is an enzyme that depends on copper and zinc in its active site. But it's, this specific structure actually contains cobalt. And this is quite common, commonly seen in the place of copper. So actually the annotation says it is a modified physiological site where the relevant metal ion has been substituted by another one, which has been used in this case, presumably for as a spectroscopic probe as well, since cobalt is an MR active. So it's been put in place of the physiological zinc ion. And again, the small book icon will point to the relevant literature to support our assignment. And that's one of the things we've been working recently, as I mentioned, and we hope to be able to push this to the PDBE via the PDB knowledge base, so that in their pages, this annotation about the physiological relevance of metals will be available, distinguishing therefore spurious metal ions or metal ions that have been, let's say, added for the sake of their anomalous diffraction and things like that. Regarding other contents in Metal PDB, which will also be useful later, one of the things is we build metalloprotein families based on their sequence similarity. And one of the things that we think is useful is in this context, at least for people who are looking at structures rather than solving them maybe, is that we have the opportunity to divide the structures that belong to any specific family based on its metal content again. So we can, at the top of the list, this is again for carbonic anhydrase, so a zinc-based enzyme. At the top of the list, we have all the members of the families that have a structure containing zinc in the PDB, so they're very similar to the physiological one. The second group is structures where the metal binding site is conserved. So in all these alignments, we check that the ligands to the metal ions are still present in the sequence. But the site is binding a different metal. So this is the metal replaced subgroup of structures. And as you see there, and it can be different metals. There's copper and cobalt and mercury on this list. And this is not the same mercury as before. This is mercury that has been selectively substituted in that specific site. Then there's structures where the metal is missing. So you have the amino acids that bind to the metal are still present in the sequence, but the metal is not in the structure. So that can be a mistake or can be an experiment where people have crystallized the protein in absence of metal ions by depriving the protein, for example, with EDTA. And finally, at the bottom, we have proteins which have enough sequence similarity to be included in the family, but they are actually lacking the amino acids that bind to the metal. So presumably this is an evolutionary or maybe a design where the metal site has been lost. Of course, we provide statistics, so I can check whether this, for example, a new structure is similar to what's already in the PDB. These statistics are relatively high level. So, for example, we don't discriminate with respect to resolution in these plots that are automatically generated on the web pages of the database. So you can see the most common coordination geometry at the top, the most common ligands in the center, and the range of typical coordination distances that you see for different atoms. Okay, so that's about the contents of the database, and then we try to come up with some tools that would make it more useful for the communities. And one is this metal S square that stands for metal site superposition. And it's a way to superimpose the structure of two metal sites without looking at the fault. It's relatively common, and I have a couple of examples in the next slide, that you can find the same substructure binding a given metal ion in quite different folds, so evolutionarily independent folds. And then we wanted to compare these substructures in an unbiased manner, and especially looking at the metal itself. So we came up with this algorithm that is just a variant of the algorithm you normally use for structure superposition, which actually starts from superimposing the metals and then the amino acids that are coordinating the metal ion in the site, and then we'll optimize the rest of the site. And it never looks at the entire protein fold. It's an entirely local superposition, and this allows us to look at sites, as I said, embedded in different folds, as you see in the small example in the bottom left corner, and recognize whether they are locally similar or not, by making sure that first of all, the metals and the metal binding amino acids are properly superimposed. So a couple of applications. For example, you can automatically cluster sites. This is done within the same families, where we can actually identify different subfamilies, let's say, at the structural level, by looking, for example, whether you have a substrate in the site or not, or whether you have inhibitors of different size with different binding modes. These are very straightforward clustered based on these analysis of just the local sites. And another application is, as I mentioned, we can look at sites independently of the folds. So this is an analysis of iron sulfur binding proteins. Iron sulfur clusters are small clusters made up by 1 to 4 iron ions bound to cysteines or histidines and sulfide ions. And so you can see that you can take more or less 50 different folds and actually cluster them using the algorithm I just mentioned into just 4 groups based on the local similarity. And this is also for other systems. We actually initially observed that by looking when looking at zinc fingers. So it's, we believe, a relatively common feature. Now, I just want to mention a paper that's been published by a completely independent team, where they reanalyzed by using PIXI 32 structures that were deposited in the PDB by the Structural Genomics Initiative in the US. And they checked using PIXI whether the metals that they saw in the samples, so they produced the sample anew, matched the metal that was present in the deposited PDB structure by the Structural Genomics Consortia. And essentially, they found that about 1/4 of the proteins, of the structures, sorry, contained metals that were not consistent with the PIXI analysis. And again, I took from there, so this is just appeared in JAX. And again, I took from their paper an example where they have, where they compare this 3GGL structure, which has been deposited with zinc in the metal side by the Structural Genomics Initiative. And from my reading of the paper, I understand that they found that 3 other metals were possibly bound in that site, but definitely not zinc. And they actually proposed to remodel the structure with manganese, and it's available in the PDB with this new code. So I thought, how can metal PDB help in such a case where you actually have experimental data that tells you some options about which metal is in the site, so you don't have to guess entirely, but you still have choices to make. So I took this very same family, of course, and I looked at another kind of group or clustering that we have available already from metal PDB, which is a fold comparison. So we take all proteins or structures of proteins with the same fold. We do a regular structure superposition of the fold, and then we check whether we have metal sites occurring at the same position within the fold. But we don't care about which ligands are there, so which amino acids are binding the metal, which we don't care about the identity of the metal. So it's just a way to look at which sites are present in a given fold family. And we call them equistructural sites in in our jargon. So if you do this analysis for that protein family, you actually find besides 3GGL no other zinc binding structures, but you don't find manganese binding structures either. And actually, the majority, nearly 3/4 of the sites that occur in the same fold at the same position are binding calcium in their binding sites. And there's a group, the second most common metal in that site has been deposited as sodium, which I didn't look into it, but doesn't seem very likely to be correct. Let's put it that way. So I would say that actually, if you are given a choice between calcium, manganese, and iron, on a very simple reasoning that probably your the structure, the metal that's more commonly found in that fold is also the most likely candidate for yours. And since it's in agreement with the experimental data, would be calcium. Naturally, you can look at the superposition of these various sites, and you see that they are all identical. So 3GGL is the original structure versus one of the calcium binding structures in that cluster. And you see that the superposition is very similar, although the beta sheet is slightly different in the zinc binding structures. If you take the refined structure with manganese that's been deposited last year, again, it's essentially identical, and you get a better view of the beta sheet. So it tells you that anything that you put in that site is actually reasonable as long as it agrees with the data. And just briefly, I want to mention another tool that we use. So we have a database, we have an algorithm to compare pairs of metal sites. So of course, the next thing we did was to make it into a tool to browse the database. So now you can upload a new PDB structure. The software will identify the metal sites in your structure, and we go through the metal PDB database and find all sites that have local structural similarity to the metal site in your newly solved structure. And so again, this can give you a hint which are the structural features around that metal that are already known from the PDB in a way. And you can actually also discover some new relationships. So this is from a publication using the tool actually, where they discovered structurally similar binding sites with different metal ions loaded in the structure, but fairly good local structural similarity, as you can see from the superposition. This was a little helped by the fact that this was an all helical protein. So in the interest of time, I'm just going to skip the next 2 slides and thank you all for your attention.